Our talk for today is about why we need sleep, and our speaker is an expert in the molecular mechanisms of sleep. Professor William Wisden is the Chair of Molecular Neuroscience at Imperial College London. He studied natural sciences zoology at none other than the University of Cambridge, and then did his PhD with Professor Stephen Hunt at the MRC Molecular Neurobiology Unit, Cambridge, followed by a period as a postdoc in Professor Peter Seeburg's lab at the University of Heidelberg, Germany. Professor Wisden has worked extensively on neurotransmitters and the neural circuitry of memory. Most recently, he became interested in sleep. In collaboration with Professor Nick Franks, also at Imperial, he used mouse genetics to investigate how inhibition regulates the sleep-wake cycle and the actions of sedative drugs. Professor Wisden is a fellow of the Academy of Medical Sciences. Without further ado, let us invite Professor William Wisden to give his talk on why is sleep good for us? Professor Wisden, please.
Thank you. Can you hear me all right, Bram? I'm
yes,
sounding
perfectly.
okay. Thank you. And I'm guessing I can't see anybody except I can see my slides. So I'm sort of talking into a vacuum, but I guess you're all listening. So thank you so much for inviting me, Bram, to the SciSoc up in Cambridge. And I'm sorry I can't physically come up to Cambridge. It's always a place I like to visit and have a glass of sherry or whatever you do after the talk, or go for dinner to meet some of you. So that's how life is at the moment in our strange virtual environment. So in the next hour, I'm going to tell you something about sleep and a bit about our own research. So we all know what sleep is. You can tell immediately if someone's asleep. Like if you look at any of these classic pictures here, you immediately see, well, someone's asleep. And we can tell if someone's awake. It's just sort of hardwired in our brains to recognize what sleep is. But if we wanted to be scientific about it, I guess we'd say that sleep's a reversible behavioral state of perceptual disengagement from and unresponsiveness to the environment. So that's a sort of formal definition, but we all know what that means ourselves, since we sleep every day, hopefully. But sleep's widespread in the throughout the animal kingdom, and it's not just we mammals that sleep. Birds sleep, of course, fish sleep, and jellyfish and insects. And this is actually a picture of a moth I took on holiday. I noticed just with my phone it was awake, and then a few hours later it was still in the same place. This is in Corsica, fell asleep on this tree trunk. So insects sleep. They have a period of quiet, at least, and there's a lot of research on studying how insects sleep using the model organism, the fruit fly Drosophila. But I'm, of course, going to talk mainly today about experiments that we do in mice. But I show this slide simply because sleep, or rest at least, seems to be a universal phenomenon in animals. And as Bram mentioned, insomnia, not being able to get to sleep, it's a very common problem. I mean, most of us from time to time, I'm sure, always have difficulty going to sleep. I'm actually myself a very good sleeper, but even if I get stressed about something, then it does keep me awake at night, and I'm sure it's the same for you. And recently, Tracy Emin, as an artist, she made an installation in London, I think it was last year, where she's a terrible insomniac, and she photographed herself with her phone in all the different, throughout the night, in all of the different expressions she has on all the different days. But as I'll tell you later, so strangely with insomnia, if you actually measured someone's brainwaves, which I'll explain shortly, you actually find that insomniacs actually do sleep, and they have the same amount of sleep often as people that are having healthy sleep, but their sleep is fragmented. Anyway, but I'm really merely showing this slide to show it's a common problem. And, you know, there are many more critical problems with sleep in society. So again, as I'll explain shortly, it's actually a form of torture if you keep somebody awake. There's actually a pressure to go to sleep. But in the 1960s, this is somebody tried actually for the Guinness Book of Records. It was a teenager, Randy Gardner. That's this young man here. He's still got the current record for the longest anyone has stayed awake, and it was a pseudo-scientific experiment. It wasn't actually done where they looked at his brainwaves to prove that he was awake, but he had friends and some scientists trying to keep him awake. And this was a press phenomenon at the time in America, and everyone was very excited about it. And apparently afterwards, he just slept for a couple of days and then woke up and apparently had no other problems in life. But this is all rather anecdotal. And you're not really allowed to do that experiment anymore. And it's quite likely that, in fact, he did sleep in micro sleep, but this was not scientifically recorded. And then, of course, maybe I'm showing my age here, but from these kind of old rock musicians like Keith Richards from the Rolling Stones, they took lots of stimulants. And with stimulants, you can also artificially stay awake for quite a while, but eventually the pressure to sleep is overwhelming. But more importantly and more realistically, for people in everyday life, sleep deprivation often comes from parenting. So that's one of the biggest challenges to people in your normal life. If you ever become a parent, you have a massive amount of sleep deprivation. And for whatever reason, if you don't sleep, you can't, there's still a pressure to sleep. And so people fall asleep at the wheel. So this is a very significant problem. And sleep apnea is a phenomenon where it's actually got, it tends to be for overweight people where when they're asleep and snoring, their windpipe gets pressed shut, and then the brain senses a lack of oxygen and wakes up the person. But so that's very unhealthy, but it's got nothing to do with sleep per se, although it does give the partner of that person, if they have a partner, they also get sleep deprivation. And interestingly, some people don't need much sleep at all. I've got a slide of that coming up in a minute. And rather, only maybe some of you are in the audience, you don't need much sleep. Some people apparently only need 4 hours. And a Prime Minister, Margaret Thatcher, she was Prime Minister when I was an undergraduate at Cambridge, actually. She famously needed only 4 hours sleep. And here's her husband, Dennis, fast asleep at some political conference. Interestingly, Margaret Thatcher went on to develop dementia, but we don't know if that was related to her lack of sleep. And again, there's more on that later. So insomnia, it's a plague for people. And there's no end to what the public will go to. They'll try anything. People will try anything if you don't have a science background in order to sleep. For example, I mean, this is just an advert I took from the Times newspaper a couple of years ago. And here we have a Philip Stein sleep bracelet designed to boost melatonin production for £265. So this complete kind of fake science, or not even that, but just all kinds of things. I think one of the most useful things here is simply the eye covers. But many of these other things, of course, a good mattress is important. So moving on to the science, what is sleep? So we've already looked at that when we looked at the paintings, a reversible behavioral state of perceptual disengagement from and unresponsiveness to the environment. So that's, if we wanted to be scientific, that's what we would say sleep was. Obviously, you're not conscious, but it's not like being in an anesthetic state, which is something we also work on in the lab, but I'm not going to talk about today. So you're not anesthetized, but you're definitely not aware of the environment when you're asleep, and you're harder to arouse. I mean, if you're in a deep sleep, you'll know that from friends or family, you're going to have to shake somebody in order to wake them up. And you have particular postures. depending on the animal or us humans, we curl up in particular ways. It's very obvious that we're asleep. And what I'm coming on to now, there's a way of looking at the brain waves through the electroencephalogram, the EEG, to actually quantify how our brain is sleeping. And this all started with a German scientist and medic called Hans Berger around last century at the time of World War I. So around 1917, 1918, this is a well-known story. He was a horseman, a cavalryman in the German army, and his sister in Berlin had a dream that he fell off his horse and seriously injured himself. And this turned out to be true. And Hans Berger got her letter when he was still fighting, and she'd had a dream that he'd fallen off his horse and broken his leg or something like that. And he was so shocked by the coincidence, he decided when he got back from the war, and because he was a medical doctor, he had access to research facilities, that he set up a research program to investigate telepathy, because he was convinced that his sister had read his thoughts. So that sounds like voodoo, telepathy, but don't forget this was 1918. But he was a very good scientist. So in the grounds of his hospital in Jena in Germany, he set up an apparatus to detect brainwaves because he wanted to test telepathy. And so he was the first person, he basically was the inventor of the EEG in humans. Other people had attempted similar experiments in things like rabbits. But anyway, he got that working for humans, and he measured brainwaves from subjects, and he found regular rhythms. So these are tiny fluctuations of millivolts picked up with these electrodes on the scalp. And then he realized that the signal was so small that telepathy could not exist. So he was an excellent scientist, and he disproved his theory of reading people's thoughts, but he started a new field of interpreting what the EEG means. And this method, essentially, although the electronics have improved substantially, is still used today. And strangely, nobody thought about doing it in sleep. It was actually this man, Nathaniel Kleitman. He was actually from Moldova, in the middle of Europe, but he emigrated to the USA, and he was the first person, actually not until the 1950s, to record the sleeping brain versus the awake brain. And this is a picture of Kleitman himself. So he's a pioneer of sleep research, and here he is having his EEG measured during sleep. And he's a remarkable character. Another slide coming up from him in a minute. He lived to 104, and he wrote this at the time, one of the first textbooks about sleep, and it was called Sleep and Wakefulness. And he said he put the word sleep first because at the time in the library, there was no heading for wakefulness or awake. So this was a new field, studying sleep. Although humans did it every day, somehow scientists hadn't really, with one or two exceptions, thought about studying it in terms of the electrophysiology. So there are a few exceptions, again, coming up. So that's Kleitman. And this is how we do it today. This is a colleague of mine, Joos van Sommeren in Amsterdam, and he's a specialist on measuring human sleep. And this is the modern electrode array that's used to detect brain waves during wakefulness and sleep in Van Sommeren lab and other labs. So from the work of Kleitman and other pioneer scientists, we know that when we're asleep, there are two types of sleep, and these are almost like household names, even if you don't even know anything about sleep or you're not even a scientist. But everyone's heard of REM, rapid eye movement sleep, and then non-REM sleep. And again, this is really a textbook view because this is very well established. Kleitman and his colleagues, for example, Dement, they discovered in the 1950s that as we go to sleep, our brain waves change. So when you normally look at the brain of an awake person, of us right now, it's a mixture of all kinds of different frequencies because neurons throughout our brain cortex are doing different things, and the signal although there's information in it, it's very complicated, and the technical phrase for this is desynchronized. It's a low power desynchronized signal with many different elements. But as we get sleepy, and hopefully this will happen to you tonight, there's a little, the EEG changes. So maybe let's just say this is going to happen to you at 11 o'clock tonight, that your awake EEG, as you start to feel drowsy, will have something called a spindle in it, which is a sort of ripple of a particular synchronized frequency at about 10 hertz. And then as you get more and more drowsy, your EEG settles into a 4 hertz rhythm. And that's called, between 1 to 4 hertz, and that's called the delta rhythm. And there's sleep scientists classify that into 3 stages, 1 to 3, and you descend. This is a sort of conceptual diagram of the human sleep. You descend into these stages. You stay like that in deep non-REM sleep. So that means your brainwaves are oscillating around 0.5 to 4 hertz. And then for an unknown reason, 9 to 6, maybe an hour later, your sleep gets lighter, and then you enter this fascinating state called REM sleep. And in REM sleep, your muscles are paralyzed. They're not completely paralyzed in non-REM sleep. So the technical phrase for that is atonia. So you can't move, but your cortex has an awake-like EEG. And so you come from the non-REM into this red line here, that's REM sleep, and you go through multiple cycles through the night. And then, still a complete mystery, you wake up in the morning from REM sleep, usually feeling refreshed. And we don't really know what that refreshment is. You can ask me questions about it, or I'll talk about it as we go on. So we have these 3 stages of non-REM sleep and periods of rapid eye movement sleep, and the brain cycles through these states. And so the brain is active during sleep. It's not turned off like it is in deep anesthesia. And bizarrely, if we were to look at muscle electrodes on our eyes, during this rapid eye movement sleep, our eyes flicker backwards and forwards. No one knows what that means, actually. It's probably kind of an epiphenomenon, but that's where the name comes from, rapid eye movement sleep. So you can't move your skeletal muscles, so you can't move your arms or your legs. Obviously, the neurons that control your breathing and your heart muscle, that's all still working. So you can breathe, and your heart is pumping. You can't move your skeletal muscles, and bizarrely, your eye muscles flicker backwards and forwards. The French call this, there was another famous pioneer of sleep science, Jouvet, and the French call this the paradoxical sleep, because the EEG looks like you're awake, but you're actually asleep. So we can do the same experiments in mice. So here's one of our mice, and with our collaborator, Alexei Vysotsky in Switzerland, he developed a little, what we call a neurologger. So that actually allows a mouse to have a normal laboratory mouse life, and it's no cables, and it collects the brain EEG, and then we download this onto a offline for analysis. So it's a way of collecting the mouse EEG. And when we do that, we find that, well, many other scientists before us find that mice, they have the same sleep states as we humans. So you can look at their muscle. This is the electroencephalogram, the electromyogram is the muscle, wake, the muscle's twitching, and in non-REM sleep, it's sort of a little tremor, and in REM sleep, it's completely silent. And you can do EEG analysis. And so this might all look a bit similar to you, and it does to me, but so what we do is a technique some of you might be familiar with called a Fourier transform of the data, and that turns it into particular frequencies. And then it'll become apparent with numerical analysis that this is a mixture of frequencies, and non-REM is primarily 4 hertz, and REM is actually a frequency that's predominated by theta rhythm. It's about 10 hertz, but it looks quite similar to the EEG. Anyway, the interesting thing is mice are not like us in the time that they sleep, because we have one block of sleep all at the same time. But most animals, most mammals, they sleep distributed throughout the day. So you might think, well, a mouse is nocturnal, and that is true. So right now, it's getting dark. The mouse is waking up, going out of its little mouse hole, but it's also going to have a nap in about an hour's time. And during the day, it also wakes up a lot when it's also more sleeping. So this is a technical phrase called polyphasic sleep cycle, but that's more normal. So we primates are exceptions in having one block of like 7 or 8 hours of sleep. And won't linger on this too long, but physiologists understand why we have these brain waves, the EEG, different frequencies, because the neurons fire during the different sleep states at different frequencies. So for example, I'll just show you this raster plot. These are 4 different neurons, and they fire action potentials at a 4 hertz frequency. We don't know why, what the chemicals are that make them do that, but they do in non-REM sleep fire at this regular frequency, and so that extrapolates to the surface of the brain and gives the frequency of the EEG. So one of the interesting features of sleep is when do we sleep? One is when do we sleep, and the other key question is why do we sleep? And again, this was first explored by Kleitman in the 1930s. So Kleitman was a truly original scientist, way ahead of his time. And he and a colleague, Richardson, isolated themselves in a cave, Mammoth Cave, in the US. And again, it was a sort of sensational thing to do. They got all the press involved, and they had people taking pictures of them. And so what Kleitman wanted to do was to see in total isolation how that affected his sleep and his body rhythms. But because they were always interrupted by the press and stuff, it wasn't really a great experiment. But he set the principle, and then in the 1960s and 70s, it was done properly by, amongst others, a German group led by Professor Aschoff. And so what they did was they took volunteers in a cave near Munich in the Alps, and they had the volunteers on a fixed light-dark cycle. And you can see like midnight to 12 o'clock lights were off and lights were on, and then they followed the activity of people under these fixed conditions, and then they put the lights on the whole time. And then they could see that these human volunteers, their clock started to drift. So they started to they didn't have any cues from the outside world. So they started to get up later, they went to sleep later. They always slept for about the same amount of time, but their clock drifted. And these experiments have been similarly done in animals, and it's a whole large field of circadian science, but it shows you that the part of the timing of sleep is under circadian control. And this is an important thing to take into account because, especially shift work, which is still done a lot, of course, is vital in society for doctors working shifts, or before COVID, airline crews, people running power stations or steering oil tankers. Many people are on shift work. And if you move from having a sleeping normally at night and then suddenly your shifts, let's just say I'm giving this lecture now, and then afterwards let's just say I've got to stay up all night I'm going to do some sleep experiment. And then I've decided to stay up tomorrow as well. That's the kind of thing that gives you jet lag. So some colleagues at the University of Surrey, led by Simon Archer, the University of Surrey is one of the few places in the UK with a proper human sleep lab. So they take mostly undergraduate volunteers, and they pay them, and they put them in a proper sleep physiology lab, and they give them sleep regimes or sleep deprivation. And in this experiment, they delayed allowing people to go to sleep by 4 hours for 3 consecutive days, and they measured their blood, the blood cells, the white blood cells, gene expression, and they found an amazing change in gene expression of many of the whole genome, actually. The whole genome changed its expression quite substantially. But interestingly, there's a chemical, a hormone released into our blood when the lights go off. And this melatonin, that stayed the same. But so the melatonin stayed in phase because that's determined primarily by whether it's light or dark. But all of the many key genes involved in cell replication, translation, transcription, all kinds of stuff became out of phase, which doesn't sound like that would be too healthy. So that could explain, for example, if you're doing a lot of shift work, why you could get a heart attack or get fat, even if you're eating normal amounts of food. So the circadian thing is one thing controlling sleep. And then there's the thing that we work on, which is the homeostatic drive that influences sleep intensity. So the circadian drive tells you when in the day you're going to sleep, or an animal is going to sleep. Is it going to sleep at night or the day? And then there's this thing called the homeostatic drive, which is actually all about catching up on lost sleep. And it's something to do with tracking the time awake. And it's got something to do, we think, and other sleep scientists think, with the function of sleep. So the circadian drive is all about the ecology of the animal and us. Are we designed, have we evolved to be active in the day or the night? When should we sleep? And the homeostatic drive, as I'll explain, is about the intensity of sleep and catching up on lost sleep. So the body makes an effort to catch up on lost sleep, as I'll explain. But just to finish off on the circadian aspect, there was a very famous experiment shown here in this paper done by Stefan and Zucker in the early 70s, and they basically located the place in the body which has the clock. And that's here. I apologize for this crude diagram, but it makes the point. This is a rat brain. This is the front. This is the olfactory bulb. This is the back and the spinal cord. And they found that there was an area in the base of the brain in the hypothalamus called the Suprachiasmatic Nuclei. And for a variety of other evidence, they suspected it was the clock, and they simply lesioned it. They cut it as a very crude but straightforward experiment. And here they got a sensational result. So this is a control lab rat, and they're looking both at how much it's walking around, so that the converse, of course, is it's sleeping, and how much it's drinking. So it's its activity, A and D. And you can see, without really even analyzing the data very carefully, that being active and drinking happens more in the dark, and in the light there's less so. Whereas when the rats had their this part of their hypothalamus lesion, there was no pattern to it. So they were active and sleeping and drinking randomly. So this is very classic textbook work showing how the clock controls the pattern of when we sleep. But you still sleep. And subsequently, these three scientists, and I'm sure some of you will have come across this in your lectures, these three scientists, Jeffrey Hall, Michael Rosbash, Michael Young, they were given the Nobel Prize for working out the molecular mechanism of the clock, which is a universal thing, actually. So that's found in plants, Drosophila, us, and all our body cells turn out to have a clock. And these three scientists and their colleagues worked out the mechanism. There's also, I should say here, there's a very well-known circadian sleep researcher in Cambridge down at the LMB, Michael Hastings. So I think you should have him along at some point as one of your speakers, because he's very engaging. And he's got a good video of his own brain showing the SCN. Anyway, I diverge. What is known now is that there's a molecular mechanism for the clock. And when you destroy this genetically, here's a normal mouse. It's active and sleeping. This is its activity on a wheel. When you get rid of the clock, it sleeps and is active randomly. So it's still sleeping. And when you sleep deprive it, it catches up on lost sleep. So that's the circadian. And nevertheless, there's quite a few genes, we don't understand, to be honest, how any of that works. But some of us are larks. I'm a lark. I always wake up at 5:30. When I was young, I used to work late at night in the lab and I studied late. But somehow in my late 30s, early 40s, I switched and I became a lark. I can only work properly now in the morning. But many of you, especially younger members, will be owls. So you'll actually be waking up right now. You might be doing your best work later this evening at 10 p.m. So we know that this is partly genetically controlled, and you shouldn't feel bad either way. Whatever you are, you are. You just have to go with the flow of your what's called your chronotype. But we know that there are many genes that regulate this. And from rare human families, some individuals always wake up early. Some people have delayed sleep phase disorder, and some people are short sleepers. And this is always turns out to be mutations in some aspect of this clock mechanism, but we don't understand yet, the researchers working on this problem, how it feeds into timing sleep. But nevertheless, it's clearly a lot of this is under genetic control. There's also, going from the other extreme, that some animals sleep because they're at the top of the food chain. That's the hypothesis. They haven't got any, so although they've got a core need, a certain part of their sleep is essential. This leopard spends, or lion, or like my pet cats, they spend most of their time asleep because they haven't got anything else to do. They're just designed to eat, kill some prey, and then they've sort of evolved to have extra, this is my opinion, extra sleep just to fill in the time and stay low to save energy. But if we were to sleep deprive them, they would still catch up on lost sleep. So there's an ecological aspect to what I would say is excess sleep. And we can broaden that out a bit. So I've been talking about sleep debt, and I'm going to say a bit more about that. If you've got a high sleep debt, you need to catch up on sleep. That's sleep homeostasis. I've just spoken about circadian timing, whether you're asleep or awake. But there's a whole load of other interesting features: metabolic state, immune function, predation risk, mating opportunities, seasonal and migratory status. So birds can suspend their sleep for some species of birds while they're migrating. So sleep can be overruled. So, but still, from my perspective, coming to what our lab works on, you can do all these genetic experiments on mice, and there's actually literally hundreds, if not, I would say hundreds of mutations in mice which have been made deliberately or randomly in their genome, which have affected the timing of sleep, like the circadian timing. But no one's so far, I might be proved wrong, but so far no one's ever made a mouse that doesn't sleep at all. So sleep must be vital. It's somehow as important as breathing, but actually, as Bram said at the beginning of this lecture, sleep researchers can't agree on the purpose of sleep. And sleep seems so important that a dolphin, for example, Jerome Siegel's lab has shown that there's alternating sleep on each side of the dolphin brain. So the dolphins are swimming all night. I think it's the same with whales. They can't afford to stop swimming. So one side of their brain goes to sleep while the other stays active, and then it alternates. So this is what we work on, which is this fundamental aspect of why is sleep inescapable after prolonged wakefulness. And our lab and other researchers think that this is a fundamental drive. It's it's as important as hunger and thirst, and all of these things become overwhelming if you haven't eaten, if you haven't drunk anything, or if you haven't slept. Depending on the length of deprivation, there's an overwhelming drive to eat, to drink, to sleep. And these things you would do anything for if you were hungry enough, thirsty enough, or sleep deprived enough. It's an amazing drive, and it overcomes the circadian rhythm. And some of these things are specified in the base of the brain at the hypothalamus. So we're working on this problem in red. What's the sleep drive? And what are the physiological reasons for sleep? So strangely, sorry for the misspelling here, we can't agree. We don't know. But this illustrates the power of sleep deprivation. So sleep researchers, they think of sleep, controlling sleep as two drives. One is the circadian, and that's shown with this red sine curve here. And as the day progresses, some kind of urge specified by the circadian clock increases, and when we go to sleep, it decreases. And then there's something called process S, which is the longer you're awake, the stronger the urge to sleep is. And also that decreases during sleep. So and this is what we've tried to find in our lab, what process S is. And you can override it. So if I said to any one of you now in my audience, which I can't see, well, what's today, Tuesday, if you can stay up till Sunday and I measure your EEG just to prove that if you can stay awake, I'll give you more than, what did Bram say, a microphone. I'll give you £10,000 if you stay awake till next Saturday without taking any drugs. And some of you might agree to that. And you can actually, to some extent, this is called top-down control. You can override, let's say your goal, work late for money. You can override the homeostatic urge to sleep, which is increasing and increasing in this blue line. And then at some point that just collapses. What's today, Tuesday? It probably just, your will to sleep will be so strong, even though you want to stay up and win that bet, that you'll just pass out, and I'll easily win the bet by Friday, unless you're taking stimulants or something. So that's an amazing thing. And what actually controls that? So the short answer is we don't know, but we can measure it in the lab with our mice. So you can give mice sleep deprivation with novel objects. So if you put a novel object in a cage, at the time the mouse is feeling most sleepy, it sort of goes, well, that's interesting, I'll stay awake and explore it. And another hour later, you give another object, like here it's a marble, it says I'll stay awake and explore it. And here you give another object, another marble, stay awake and explore it. And if you measure its EEG, this is the sleep deprivation, it doesn't sleep. And then when you put it back into its cage, it has a huge amount of non-REM sleep, the first state of sleep. So that's called recovery sleep technically, and it'd be the same for us if you were up all night at a party and then you crash out in the morning. So somehow that phenomenon illustrates the importance of sleep. So after sleep deprivation, we catch up on lost sleep. So sleep must be somehow important, otherwise why would we catch up on it? And our lab, I'll show you a slide in a minute, is developing a new brain circuitry for this sleep homeostasis. But before that, I want to go just briefly mention another pioneer of sleep, which is von Economo. And he was an Austrian scientist at the time of World War I again, like Hans Berger. And at the end of World War I, in 1918, there was, you'll probably have all come across this by now, because of COVID, the Spanish flu pandemic. So not COVID, but Spanish flu, where millions of people died, and it was all over the world. And some of these people survived, but had brain damage. And von Economo was a Viennese, an Austrian neurologist who had certain groups of patients that slept all the time after having Spanish flu, and certain groups of patients that were awake all the time. And he published a paper on it. When those people died, he did postmortems of their brain, and he found that one lesion of the brain, at the bottom of the brain in the hypothalamus, caused people to stay awake all the time, and another lesion caused people to go to sleep. If you click on this YouTube video, you'll see some images from the Wellcome collection of these patients. So here we're talking about the base of the brain. The Spanish flu had done some damage to the brain, and it affected either wake centers or sleep centers. And nowadays, we know from work of colleagues such as Professor Cliff Saper at Harvard University, there's a network of neurons which are keeping you awake, releasing different transmitters. This is the human brain. So when you're awake, there's a collection of neurons releasing different chemicals, such as noradrenaline, acetylcholine, and histamine into the cortex. And that's somehow keeping us awake. And when we go to sleep, there's a special class of neuron which makes an inhibitory neurotransmitter, a chemical called GABA, and it switches all these off. That has been well documented in mice and rats. But the question is, we still don't know what drives the switch. Why do we go from awake to sleep and sleep to awake? What happens when you're sleep deprived? And we don't understand how the circadian clock talks to this mechanism. But this is these essentials here are well understood. So in our lab, for example, we've discovered, I just mentioned this briefly, several new types of neuron that regulate catching up on lost sleep, because we do mouse genetics where we selectively kill different types of neuron in the brain, and then we measure how we do these sleep deprivations, and we measure if the mice can catch up on lost sleep. So this is a mouse where it was given sleep deprivation, so it loses its sleep, cumulative sleep loss over 6 hours, and then a healthy mouse catches up on its lost sleep when we put it back to sleep, whereas mice missing certain classes of neuron, and I'm not going to explain precisely what these are because it's in a detail really for this talk, they don't catch up on lost sleep. And we found a whole collection of these, so you don't need to understand this diagram in detail, but we found a whole collection of novel neurons around the brain that sense sleep loss and then get overactivated when the mouse is being sleep deprived, and contribute to putting the mouse to sleep. So it's as if different types of neurons in different parts of the brain can sense sleep loss and then put the mice to sleep. So we don't need to linger on this too much, but we don't understand why these neurons get activated. So sleep scientists, there's something called the Somnogen Model. So we look for some chemical that activates the neurons when we get sleepy. There's no consensus in the field about what that is. Some people say it's adenosine. Some neuroscientists, based on work in Drosophila, the fly, say that special ion channels. And some scientists say it's a mechanism like memory, that synaptic strength changes while we're getting sleep deprived. But in fact, there's no consensus. And we haven't found, we're actually working on this mechanism here. So the other thing is that as we get sleepy, we adopt specific sleeping postures, whether we're a human or a cat or an otter. And Ed Harding in the lab, he's now working on the Addenbrooke site, has discovered that warming the skin actually induces sleep. And he worked out the mechanism for that, which goes from sensory neurons in the skin, which send axons to the brainstem, and they release a transmitter, glutamate, and they go to the hypothalamus, and they both induce sleep and body cooling. Now I realize, actually, because this is a strange virtual format, I'm going to run out of time because I've been too relaxed sitting here at my desk telling you about sleep. So I'm going to miss a couple of slides, which is not very professional, but I'm sorry about that. Otherwise, I'll speak too much. So what Ed found, I'm just going to miss out a bit, is that skin warming induces both sleep and body cooling. And what that means is when you go to sleep, your body cools down. But in order, paradoxically, for that to happen, you have to be warmed up. And so this is actually called the hot bath effect. So that's actually wrong. When you see in popular accounts of sleep, the idea that you must be cool, that's wrong, actually. You don't need to be cool. You should actually warm your skin before going to sleep. For example, with a hot bath, and there was actually about 10 years ago a paper in Nature that if you wear bed socks especially in older people, it induces sleep. And so this actually been worked out now by Yusuf van Sommer in Amsterdam. So warming the hands or feet induces sleep. We think this is the actual mechanism. But it's interesting that the body has to cool down during sleep. So although you need this warm skin to induce sleep, for some reason, when we don't know the answer, you actually cool down during sleep. So that may be something to do with the function of sleep. It's hardwired. And the other thing we're doing in the lab is randomly, I suppose we don't have a grand hypothesis for this work. We're just looking for new neurons that regulate sleep. And Zhao Yu in the lab, he found just by randomly searching the brain with a technique called chemogenetics, a class of neurons in the ventral tegmental area which induce wakefulness. So we just looked randomly throughout the brain, and he found a group of neurons which very potently keep you awake, just in this one tiny area of the brain by looking at the EEG. And in the human brain, this is a tiny little area in the middle of our brains, and this area is famous for making dopamine, but it's also got a class of neuron in it called glutamate, which keep you awake. And I just put this slide here for you because this shows you another important technique we use in the lab, which is called photometry, where we look at the calcium signal in neurons with a reporter protein that becomes fluorescent when we shine light on it in the presence of calcium. And we found by comparing the activity of these neurons with the EEG that they were selectively wake and REM sleep active, but they were inactive in non-REM sleep. So in that tiny little brain area, the VTA, we found another amazing class of neurons which are inhibitory. They make GABA. And again, we found these by accident, but they gave us the strongest effect on sleep we've ever found in the brain, and other colleagues have ever found. So when we just ignore this diagram, it's just a way of artificially activating just those neurons in the brain. We can make the animal sleep for 7 to 8 hours continuously. So that's a very potent artificial sleep just by activating that one type of neuron. And our current research is trying to work out the wiring diagram. How does it talk to all the other neurons? And again, just to remind you where that is in the brain. And when we selectively kill those neurons, the mice are awake for 4 hours more during the day, during their day, which is the lights off. So they're actually permanently lacking sleep. They're sort of a model for what happens when there's no sleep in the brain. And so also have researchers in the lab trying to find out if there's anything wrong with these mice, because they have 4 hours less sleep a day, which is quite a lot. And that's a permanent effect for the rest of their lives. So we're sort of aging these mice and seeing if that makes any difference to their health in whatever way, whether it's brain or rest of body. So we don't know why we sleep. And there's many theories, and you can look offline. There's this very nice review by Bringman. Scientists can't agree if it's for energy expenditure or rebalancing synapses or something to do with metabolism. There's no consensus, amazingly, in the field. But all I can tell you is the most striking observation is that nobody ever made a mouse that doesn't sleep. So sleep's really important. Otherwise, surely for a lab mouse, there'd be one that didn't sleep. But one thing we're working on in the lab is, does the brain clear toxins? And this is my last experimental section, I think. So the brain's full of blood vessels. There's an amazing density of blood vessels, which when you're learning about the brain, you don't really think of. When I studied the brain as a student, it never occurred to me there was all these blood vessels, but there's millions of blood vessels in the cerebral cortex. Obviously, it's essential metabolism, providing oxygen. And these blood vessels are serviced by glia. Here's the neurons, and here's the glia. And the end feet wrap around the blood vessels. And there's a very influential paper from a Danish scientist called Maken Nedergaard. She had a very important paper in Science a couple of years ago that she proposed a mechanism for how the brain cleared toxins, which is called the Lymphatic Hypothesis. And she discovered that toxins were emptied from the brain at the gap between the blood capillaries and the end feet of the glia. And she showed in her paper that during non-REM sleep, this space expanded and there was more flow of substances out of the brain, actually not in the blood vessels themselves, but alongside, and that's called the periplasmic space. So this is supposed to increase selectively during non-REM sleep. It's been a very influential paper in the field. And of course, one of the obvious things is do plaques and tangles, which happen in Alzheimer's disease. So these are somehow toxic deposits of proteins that build up during lifetime and somehow kill neurons. Are these actually cleared during sleep? And again, there's many well-known scientists in Cambridge working on plaques and tangles and Alzheimer's disease, but we're particularly interested in whether these toxic plaques and tangles get cleared by sleep. And a well-known Alzheimer's scientist, David Holtzman, has shown both on human volunteers and in mice that during the wake period, the levels of beta amyloid increase. And then during sleep, in the cerebral spinal fluid, they go down. So the question is that a circadian effect? Or is it due to sleep, clearing it? Or is it just due to the fact that the amyloid gene promoter is more active? So it's kind of unresolved. But this is what would be actually happening. You'd have early wake, and then at the end of the day, we're getting more toxic products deposited, and then somehow, according to the Nedergaard's hypothesis, these get cleared. And they're cleared from the extracellular volume of the brain. And if you do this chronically over a lifetime of good sleep, that would clear the brain. So I haven't got time to explain all the complexities of this, but there are all kinds of mice developed by colleagues in Cambridge, but also here in Japan, where Alzheimer's is modeled, and these toxic deposits of amyloid beta are deposited in the mouse brain, and you can see them here. So there are all kinds of these mouse models, but we're taking this particular one, mouse model, and seeing if the plaques that get deposited in its brain, do we get more of those while if the mouse is sleep deprived? And we've got two models, and this is a mouse model, which again, I'm not really going to explain. It would take another half an hour, but we've made a mouse model of insomnia. And I told you at the beginning of the lecture that Tracy Emin slides. So insomnia, actually, if you have insomnia, you do sleep in the night, but you think you don't, but you actually have fragmented sleep and wake if you do it on human volunteers. And we've discovered by accident there's a particular type of glutamate receptor in the bottom of the brain in the hypothalamus that if you lesion it genetically, it makes insomnia. So this is a normal mouse. It goes from these green things show sleep and wake, and then without showing you any statistics, in this mouse, it's got the same amount of sleep and wake, but they're all broken up into small fragments. So it's an insomniac mouse. And the bottom line is, does this cause plaques and tangles from Alzheimer's disease to appear more rapidly as we age the mice? So that's one of the things we're doing. So does chronic insomnia increase the formation of amyloid plaques? That's an ongoing experiment. And we're also doing that one I told you about just now, where we've lesioned this special type of neuron in the ventral tegmental area. There's a little bit here in the human brain, here in the mouse, where this mouse sleeps 4 hours less a day. So it's not a clinical insomniac by the technical definition of insomnia, but it definitely doesn't, it has much less sleep permanently. So what does that do to its brain neurochemistry, and does that affect it having more plaques and tangles? I've really finished my talk almost, Bram. I'm sorry for going on a bit. Is it okay, or should I stop?
No worries about that. We have a bit of time.
All right. So finally, I just want to mention REM sleep, which is that, REM sleep is the most fascinating but mysterious stage of sleep where your muscles are paralyzed. You have dreams. But you also dream a bit in non-REM. But Freud, he didn't know about not REM sleep, but he was a big fan of dreaming. And there was a whole beginning of the 20th century, people understood or hypothesized that there was a whole science of the interpretation of dreams, which Freud wrote a book about, and that shaped a lot of early neuroscience. And Hitchcock and Dali and all these kind of interesting early 20th century people were inspired by the theory of dreaming. But now we've got a more restricted view of dreaming. Nobody really accepts the Freudian hypothesis anymore of dreaming. So sensory input's blocked. You can't move your muscles, but still it's a fascinating state where you have kind of chaotic, sometimes coherent dreams. But why? It turns out that a whole cohort of researchers have discovered that if you have less REM sleep, you're more susceptible to trauma. In fact, if you have post-traumatic stress disorder, so something really nasty has happened to you, often these people have less REM sleep. And there's a whole lot of research to say that if you have REM sleep, more REM sleep, it helps you process and slowly degrade the emotional content of unpleasant memories. So my last experimental slide is this one, and this doesn't do justice to what we discovered. So we've actually, some other sleep scientists have discovered the circuitry that makes your muscles paralyzed during REM sleep. But we've discovered a whole chain of neurons, and that's shown here. We don't need to go into the details for the purpose of this talk, but we've discovered a whole novel group of neurons, which from the calcium signal, this photometry technique, they're selectively REM active. And Wade Barr in the lab has found a way of selectively silencing these neurons, which is called the lateral habenula, and it actually eliminates REM sleep in the mice. And what Way has found, amongst other experiments, is this affects the way the mice perceive fear. So normally if you have a mouse or a rat, or indeed us, who are presented with this maze, if you're fearful, you tend to spend more time in this elevated maze in the dark. But a less fearful mouse goes out in the light. So if you do REM sleep deprivation, the mouse spends much more time feeling anxious. So this supports the earlier work, this novel circuitry in humans. And finally, Brian in the lab, he's an electronic engineer, and we've got this kind of clever apparatus to switch REM sleep to non-REM sleep. I won't explain it other than the concept of what we're doing, because we want to really test the role of REM sleep. So finally, my last slides, Bram, is how not to sleep. So this applies to me, also applies to students. Light confuses the body clock, noise disturbs light sleep. We all know this. Alcohol disrupts your sleep. It only helps you go to sleep, but then you wake up. Busy mind, can't fall asleep. Hot room. Actually, it's good to have a hot bath, as I said, but not a hot room. Heavy meal, caffeine, stimulants. So all of these things, of course, are bad. But how do we get a good night's sleep? Well, it's good sleep hygiene. So you can ask me that in questions if you want, but sleep is as important as diet and exercise. You've got to take sleep seriously. You can't miss it out. So you've got to really structure it into your day, regardless of whether you're an owl or a lark. You've got to sleep. You've got to give time to sleep, and you can't cut it out, because in the long term, it's bad for your health. So finally, just a couple of career things. So I actually share my lab with Nick Franks, and Nick is actually a physicist. So he studied at King's College London, with the guy Maurice Wilkins, who was one of the people that discovered the structure of DNA. And I'm a zoologist, as Bram said, and this combination is very good. Sorry, my light in my office is this combination of working of biology and physics is very good because Nick, I think, tend to think about the brain circuits, and Nick's very good at the biophysics of analyzing our findings. And here's our lab, pre-lockdown in London. It was our last, I think it was one of our last lab socials. So I've mentioned some of the people as we went along. So it's quite a large lab. And finally, we're funded by the Wellcome Trust and the UK Dementia Research Institute. And this is a sentimental slide because I was in Cambridge, as Brown mentioned, and I did my PhD down the road at Addenbrooke's. And, you know, I was just thinking as I was preparing this talk, if I did my career, I'd still be a scientist, I think. You know, I'm very happy with my career choice of being a scientist. I might not have been a neuroscientist, but I've loved I've been lucky in my research, and I found out some interesting things. And of course, I'd worked hard, so luck and work, but I still enjoy the process of being a scientist. So I might have been in another life a botanist or some other kind of scientist altogether. And it was to some extent an accident that I ended up doing a PhD in neuroscience, because I also applied for jobs in the cell cycle and things which were topical at the time. But anyway, so I would still be a scientist again, and I've been lucky in my career. And this was a state-of-the-art microscope in 1988. It was a beautiful lights microscope, and now it looks like an antique. Okay, I'm sorry I spoke too long. It's because I'm not used to giving online lectures, really, and I didn't have any pressure. I didn't feel any audience. I think so. I probably spoke much too long because I feel I'm speaking into a void. But anyway, I'm happy to take questions if you're still there.